सायन्सची वही काढून अभ्यास करत नाही शेजारी.
प्रियंका, now get ready. Almost नहीं, अभी तो 59 हो रहे.
मैडम,
हाँ,
YouTube का ब्रॉडकास्टिंग चालू हो गया है.
हाँ,
so you can join the student with YouTube also.
हाँ, वो जो ज्यादा हो जाएंगे वो वहाँ कर लेंगे, लेकिन अभी to start with we are starting now.
ये भी YouTube का भी चालू हो गया एक्चुअली.
हाँ जी, ठीक है. Okay.
Shall I begin, Abha ma'am?
Yes, I think.
Yeah. You start
Okay.
now. Everyone, please keep your video off and also except Priyanka, na? Except Priyanka. Please keep your mobile. This also mute.
Good afternoon, everyone, and welcome to the second day of the 5-day-long International Student Development Program organized by MET Institute of Pharmacy. I'm Priyanka Joshi Jain, and on behalf of our esteemed institute, it gives me immense pleasure to introduce Dr. Anita Nair, who has been my first pharmaceutics teacher when I was studying at MET. Dr. Nair is currently a regulatory expert for API and excipient pharma registration in life sciences regulatory management at Merck, Darmstadt, Germany. From 2013 to 19, she has headed the Laboratory of Solubility and Physicochemical Properties in Central Analytics at Merck. From 2009 to 13, Dr. Nair has worked as a research associate in the research group of Professor Dr. Jennifer Dressman at the Institute of Pharmaceutical Technology, Goethe University, Frankfurt. Before she permanently shifted to Germany, Dr. Nair has been a lecturer in pharmacy at our very own Mumbai Education Trust and also at Saraswati Vidya Bhavan's College of Pharmacy. Having spoken about her professional experience, I would like to add that she has countless achievements to her credit. She has been the subject matter expert on physicochemical characterization of drugs, pre-formulation and formulation approaches in pharmaceutical research and drug development. She has the regulatory know-how on EMA, US FDA, and ICH guidance on biowaiver and bioequivalence and registration of excipients in China CDE. She has graduated the International Managerial Program in 2018 and Site Management Mentoring Program in 2017 with successful implementation of projects within the departments. She has been the beneficiary of the Pharmaceutical Education and Research with Regulatory Links European Training Network funded under Horizon 2020. She has been key account manager for cleaning validation support in biopharma and health sector in Billerica, US. She has edited and published a textbook on solubility in pharmaceutical chemistry and with De Gruyter, Germany. Dr. Nair has 12 scientific publications in peer-reviewed journals to her credit, and she has been a reviewer for reputed pharmaceutical journals like European Journal of Pharmaceutical Sciences and European Journal of Pharmaceutics and Biopharmaceutics. She has been invited as a speaker at international pharmaceutical conferences all over the globe. Her most recent lectures have been on QBD applications in Germany, BCS-based biowaivers in the US, multi-source immediate release tenofovir for a biowaiver in Australia, drugs eligible for a biowaiver in Netherlands, and standard design of pharmacokinetic bioequivalence studies in Thailand. We have invited her many times as a guest speaker for the benefit of our students as well, and they have had very interactive sessions with her in the past. So without further ado, I would request Dr. Anita Nair to present to us her topic for today, Bridging Gaps with Mini Dissolution Models. Over to you, Anita ma'am.
Thank you, Priyanka. Thank you for the very kind introduction. So I know we are almost 20 minutes into the session, so I won't take much of the time adding anything more to that. So let's start with the presentation. So as Priyanka mentioned, my topic today, what I would like to discuss with you is bridging gaps with mini dissolution models. So here we will be concentrating more on preclinical research development and how dissolution can play a role in deciding what kind of drugs move into further development, how can we evolve the drug dosage form. Before I actually get into the topic, there are 2 points that I want you to keep in the back of your mind before for this whole presentation, because that happens to be the key based on which we will see how the application works. So the first is the LADME concept. I'm sure most of you know about it, but just as a small recap, as you all know, when you take a tablet, it moves down the gastrointestinal tract. So first it hits the stomach at a particular, the pH is acidic. It will disintegrate, it will dissolve based on the pKa of the drug. And this particular step is called the liberation step. Then the drug moves all the way down the intestinal tract where it permeates into the circulatory system. So that's your absorption step. And absorption happens in the intestinal part, the proximal part of the intestine. So what you got to remember here is solubility and dissolution rate are the key factors which will decide how much drug goes into solution. You should also remember that only dissolved drugs are absorbed. So if the drug is not dissolved, it won't be absorbed. And then it moves into the circulatory system where the bioavailability concept comes into picture. That is, it gets metabolized, distributed, and eliminated, which means that the solubility and dissolution in a way is related to the bioavailability aspects of the drug, which is what every pharma company is trying to do, is to see how could they make their potential drugs bioavailable. Yeah, so this is one concept that you need to have with you. And the other one is the drug development. What happens, or what are the phases of drug development? You can broadly classify it into 3 main classes. One is the medicinal chemistry area, which is the lock and key concept, where you try to find the right molecule for a particular receptor that you want to target. Then you have the drug development phase, where you do a preclinical study, where you do studies on animals, on various other formulation. You start even looking at what kind of dosage form should I make for this particular product so that it can go into the clinical studies. Then there is the regulatory aspects, the applications, the approval, till finally the drug comes into the market. One thing that should be remembered is, in the first part is what we actually call the discovery, and the later part is what we call as the optimization process. So that is how to make that particular molecule commercial. Now, when we are going through this journey, the amount of compounds that we have in the initial part of the discovery is lots. And by the time we come to that one final drug, we have to screen several molecules before we reach here. But at the same time, the disadvantage is that although we screen a lot of molecules, the amount of substance that we have in the beginning is very less. The maximum that you can have is somewhere between 500 milligrams to 1 gram of the drug. And you need to perform so many analytical techniques before you understand what kind of a molecule am I dealing with, what are the requirements. So we are trying to generate data of an unknown molecule whose chemical structure we know, but we have no idea about its physicochemical properties. We have no idea about its solubility. So we do not have the possibility to work with large amount of molecule, which brings us to this big question: how can a mini transfer model analytic assist in the research and drug development? And that's exactly what I would be addressing today in this talk. I have divided my talk into 3 main parts. The first is I will explain to you where is the need and what are the expectations in the preclinical phase from such kind of data. Then we will look at the solution. We will see how this particular model is applied in drug research. And then we will address the question, does this model have a limitation? Is there any opportunities that we can develop with these models? Is what would be looked into. So let's go with the starting, which is the need and the expectation. So just to introduce that, we all know dissolution is something that you have heard since ages. So the first mention of dissolution was done in 1895 by the scientist Caspari, who said in the treatise that if a tablet has to be effective, it has to first disintegrate and dissolve. So this was long before we knew how important dissolution of a dosage form is. Then around in 1973, Shaw came up with the in vitro in vivo correlation, where he tried to use in vitro techniques to correlate with how a drug would behave in the human body. The biggest breakthrough came somewhere around in 95 when Gordon Amidon came up with the BCS classification. So as you all know, the drugs were classified into 4 classes based on its solubility and permeability. And then we could see how the behavior of the drug was and how the absorption would be and what are the factors that would work on it. Around the same time, Professor Dressman worked on the biorelevant media, which is now commercially available. And these media was used to understand the solubility of the drug in the human intestinal and gastric fluid. Because till then, we were doing all solubility studies. Even when I was a pharmacy student, the solubility studies were done in pH-based aqueous buffer. But our bodily fluids are very complex. It is not just a water system with some salts in that. It has much more. And the biorelevant media revolutionized how we look at solubility. Based on the Amidon's BCS classification, the biowaiver guidance came into play by the FDA. And in early 2000s, we also started using PBPK model, which is physiological-based pharmacokinetic models. These are computer-based programs into which the data that you generate on this understanding is put, and you try to predict how will my drug behave in the human body. So it is trying to see what are the risks and what improvements can I make to the molecule from a pharmaceutical or a chemical perspective. Now that being said, basically, presently when you look how drug research happens, we need to generate adequate in vitro data. We need to also have an in silico model, which is a computer-based model, as I mentioned before, into which you can put the data, and also some substantiating in vivo data. It could be in animals. Usually when it is a very new drug, an innovative drug, we have more animal model data. And if it is a generic product or the product is already in the market and you are trying to make a bridging study, or you are trying to make a new generic product, then the human in vivo data is also available. So based on these 3 aspects, you put it all together to predict what would be the best form or dosage form or product that you can prepare. Now, it is also important, if you see in the in vitro section, I have written biorelevant dissolution. So the dissolution should somehow simulate and mimic how it behaves in vivo in the gastrointestinal tract. So this is where I said you need to remember your LADME concept and understand how liberation happens within the human body. But that being said, also remember a prediction model is as good as its input data. You must have heard several times in articles, books, even in lectures, that in GMP concept, it was usually used that garbage in is garbage out. Similarly, if you put in only very nice data which you think, or which you try to adjust or fudge into the system will give you an output saying that everything is good, rosy and perfect. But that is not actually what our goal is. Our goal here is to understand how the molecule will behave and what can we as scientists do to see that the molecule gets through the development phases when it comes to biopharmaceutical based prediction. So what is the need for an in vitro dissolution so early in the research phase? So we know that dissolution is done as quality control later when the drug is developed, but so early, what can a dissolution data help us with? The first and foremost, it can help us by not discarding those potential development compounds which we think will not work well. Let me give you an example. Let's say you have a drug. So most of the drugs that are discovered presently belong to class 2. They are poorly soluble but highly permeable. Now, these poor solubility, if you do a solubility study in a particular medium, it may come out that the solubility is very poor. We even have molecules which have solubility of less than 10 microgram per milliliter. And you can sometimes disregard and reject this, saying that, okay, this drug will not be bioavailable because it doesn't show good solubility. But at the same time, it can happen that if you do proper dissolution study, which are biorelevant, you realize that this drug has a potential to supersaturate. It may show a higher concentration in the intestine in comparison to the studies that I have done in vitro. How can that happen? I will explain very soon in my slides that come up. So this is one thing that we can avoid if we have good data. It will also enable us to develop the right oral dosage form. And if required, we can use enhanced formulation. Now, what are enhanced formulation? These are those advanced formulation like lipid formulations or HMEs, hot melt extrudates, polymer mix, which can be used to enhance solubility of the drug. Again, as I said earlier, only when the drug dissolves will it be absorbed. So my main goal is either to get the drug, solubilize the drug better, or see that the dissolution rate is faster so that it can be absorbed. It also helps me to assess what kind of potential I can have with a modified release drug. And last but not the least, it reduces the need for any additional animal or human bridging studies. For example, usually if you want, initially when you wanted to generate data, you used to put the drug into the animal, try to see the PK study and predict. But remember, even an animal study is a black box. The dissolution part, what happens in the gastrointestinal tract, you cannot see. You are just seeing the PK values, or you're seeing the blood concentration values and trying to make hypothesis what could have happened in the gastrointestinal tract. But here, with such kind of simulation studies, it is possible to generate a bridge to understand the molecule better. So you can reduce the number of animal study, which is ethically good. And it also reduces cost of your research. It also increases or accelerates the time in which you move your drug in the development phase. So seeing these advantages, we should now understand what are the dissolution perspective or data information that we can get. So that brings me to this particular slide where it explains something about supersaturation. So I mentioned to you before about solubility, and I told you that the drug can behave a bit differently in vivo. It can show a higher solubility. How can that happen? We know that most of the drugs that we have or we do have in the research pipeline, they are poorly or weakly soluble drugs, which are poorly, which are basic in nature. So if you have a basic drug, you all know if it's a base, it will dissolve in an acid. And you know that if a drug has to move into the intestine, which is the site of absorption, it has to pass through the stomach. So if you look at these profile here, you take that drug, the drug will dissolve in the stomach, depending on its dose and of course its solubility in the stomach pH. And then based on the dose, it can either not precipitate. So these are those drugs which do not create any problem for us, but most of the drug have a tendency to precipitate because in the intestinal pH, which is a basic pH of 6.5, the drug shows poor solubility. Now, 3 scenarios are possible. So as I mentioned, the first one is no precipitation, or you can have immediate precipitation, or you will have a third scenario where the precipitation is in the first order and gradual. So if you see here, the drug is precipitating, but it is precipitating gradually. Now imagine this is an 8-hour window, what we are talking about. This 8-hour window is more than enough for the drug to be absorbed and to show the bioavailability, which means the supersaturation concentration is the actual solubility value that I need to feed in my computer programs rather than the dissolution or solubility data that I measure in a single medium. So which clearly states that we need something additional than the normal dissolution testings to understand what is happening to the drug when it would move from the stomach into the intestine. We should also remember there are a lot of in vivo factors that are responsible for this behavior. The first one is gastric emptying. You know that the drug empties from the stomach into the intestine in a first order phase. And this needs to be mimicked because the precipitation rate will depend on how fast the drug is coming into the intestine. The gastrointestinal volumes, you all know the BCS classification system, and the reference concentration or the volume is 250 ml. Why is it 250 ml? It represents that the 50 ml is the basic volume of fluid in my gastric compartment, and the 200 ml represents the glass of water that you take your drug with. And that's the reason we mentioned 250 as our breakout volume for the BCS classification. I need not tell you about the pH profile. Our gastrointestinal tract is having a range of pHs from 1.2 all the way to 6.8. We have bile salts in the fasted and the fed state in the intestinal fluid. The solid state of the drug that we are studying, that plays a very important role. The GI hydrodynamics, solubility, disease state, interactions, and even the formulation design will help us to understand what is happening to my drug when it moves from the stomach to the intestine. So when we look at all these factors, it is very clear the dissolution study that we have seen in the pharmacopoeia and the buffers that we use from the pharmacopoeia alone cannot capture these effects. We need something that will actually help us to get closer. Yes, it is not possible to create a stomach and an intestine like we have in the humans, but we can get closer. And that is what we are trying to see. What is required in order to move into that space. So it is now clear to us that there is a need. The instruments that we are using do not capture all these issues, and hence for a potential development candidate, we need something extra. That brings us to the solution. So Edmund Kostovich, he's also Professor Trestman's student, in 2004, he presented the transfer model. So what's a transfer model? So it's basically trying to mimic the movement of the drug from the stomach into the intestine. So he published this paper where he had or used the dissolution apparatus that is already mentioned in the pharmacopoeia. But if you see here, one compartment here is the stomach part. So you have pH 2, the simulated gastric fluid. And then you have the other compartment, which is the intestinal part, which has the biorelevant facile media. Then there is a pump in between, which tries to mimic the movement of drug from the stomach into the intestine, and the samples are withdrawn to do the HPLC analysis. So if we look at this setup more in detail, the peristaltic pump can be used to mimic the gastric emptying. When it comes to the GI volumes, the pH profile, the hydrodynamics, and the bile salt concentration, that can be governed by the media that I use and also by the paddle speed that we are using. And when it comes to formulation design, effect of excipients, you can dissolve a particular excipient within this media and try to see whether the dissolution characteristics of the drug change in the presence of the excipient or not. Well, it's a good solution, but the caveat is the volumes that you use and the size of the apparatus that we have is very large. And if you remember my second slide, I already told you we do not have so much amount of drug early during the research phase to do such kind of study. Because if I ask the medicinal chemist for 100 milligram of drug to do this particular study, naturally, I won't be one of the favorite person he or she is talking to, because they'll say it is not possible. So we had to see that we generate or we create something within our labs which can capture this information, because you already saw how important this information is and how it will help us to develop the drug. So for that, we put together a mini transfer model. So this was done, so the idea came together, and within my lab where I was leading before, we put this together and we made a very small, if you look at it, it's not very complicated. We took 2 conical flasks, made a similar setup, and the only difference is we downsized the whole model to 1/10 of the dose. So instead of using 500 ml of the media, we use just 50 ml of the media and try to see whether we can mimic a similar situation. So the advantage was for 100 milligram predicted dose in humans, all we needed to do the transfer model was a 10 milligram substance. So we could generate the data and see how the molecule behaves. But then I'm sure each one of you must be thinking, does this particular very simple arrangement match with the large volume transfer model that I showed you earlier? Because there you have nice tailored vessels with the hydrodynamics into it. We had the same question too. So we did a comparative study between mini dissolution, the transfer model, and the large volume transfer model. We evaluated the model at 3 levels, the Tmax, Cmax, and AUC. So the Tmax is the supersaturation concentration when it is reaching. The Cmax is the concentration level, and AUC was the area under the curve when that profile was generated. And as you can see here is, the values are the same. We see that when the ratio is 1 or close to 1, we see that the model is working, and the model behaved quite well at various flow rates that we used. Similarly, at the AUC, the most commonly used flow rate is at 4 milliliter per minute when it is 0 order. And then we saw that was a very good ratio between the 2 models. So that confirmed that the 2 models work similarly and they can be used. So that brings me to the next part. Okay, it can be used. So where exactly in research does this model help? So I will be now discussing with you a few case studies that we have from literature as well as some from my experiences, and show you how such a simple model can help in troubleshooting and add value to whatever we are doing. So as you can see here in this slide, it can help in the comparison of polymorphs and salts. It helps us to understand the effect of excipients on precipitation. Even the precipitation rate is an important input parameter for a PBPK modeling, and also screening of formulation. So now let's go to the first example. So this is published data of cinnarizine, which is an antihistamine. It was, the marketed products were used. And if you can see these dotted part, this is the actual in vivo data of the cinnarizine product. And here, the dark circles, they are the actual in vivo data. So what was done here was, we did a transfer model and we tried to predict the in vivo behavior using a PBPK modeling. The dissolution was done in a single unit dissolution, that is just doing dissolution in facile. And we also did dissolution by the transfer model. And as you can see very clearly here, when you just do it in one medium, we were underestimating the in vivo exposure. But when we were considering supersaturation and precipitation kinetics, we were really close to the in vivo data. So which means that, because you do have a lot of discussions in the pharma world where they say, no, the drug doesn't precipitate in the intestine. But yes, some drugs do. And because we are now doing research on these class 2 drugs, the tendency is much more higher. So this is a study which shows that this kind of models can add value and help us in creating better prediction of our molecules when we are studying through it. This is another example with polymorphism. So you all know polymorphism, chemically they are same, but physically they have different crystal habits. So then you would ask, so what? The point is, when you have two different polymers, it can show two different solubility profiles. Two different solubility profiles means two different bioavailability aspects is what we are looking. So in this example, there are two different polymers, A1 and A2. As you can see, A2 has lesser solubility, or let's say half the solubility in comparison to A1. This is the dissolution profile in FASIV, so that is fasted state simulated intestinal fluid. So naturally, the first tendency would be, let's make the formulation with A1. But you all know that polymorphism is quite dangerous if you use an unstable or a metastable polymorph in a formulation, because during storage, it can get converted to A2 and you may have to recall the product from the market. So there was a situation where these two polymorphs were existent. They even tried seeding during manufacturing with A1. So when you do the crystallization, they seeded the whole thing with A1 so as they get A1 as the product and not A2. Still, we were getting A2 as the outcome. There was a big discussion. Should we move with this molecule ahead? Should we consider it for further development? And that's where we came in with the transfer model. So as you can see here, these red lines show the profiles that I showed you earlier. This is where the solubility was at that time. When we did the transfer model, because it was a weak base, it dissolved in the stomach. And when it came into the intestine, it showed a supersaturation effect. So here, if you see, that's a supersaturation, and the concentration that we have is quite higher than what we see in the single medium dissolution study. What can we interpret from this? That there is no much difference between A1 and A2. So even if we have a mixture of these two polymers, in vivo, they will behave very similar. So it was clear that we do not have any risk of the two polymorphs. When the data was added into GastroPlus, and mind you, the data was higher than what we did by single dissolution, we saw that the bioavailability was good enough and the product can be moved further into development phases. So this is one example where it can actually help you to troubleshoot and understand how will this molecule behave, or the polymorphism effect have in the in vivo behavior. Coming to the next example, this is a comparison of salts. So when you look at this profile at the first go, it is very clear that the fumarate salt, which is shown in red, is far, far better than the parent, which is the weak base form of the drug. So naturally, the first way would be to go with the fumarate salt for your study. But what happened is when stability data were performed or stability of the fumarate salt was done, we saw that there could be a problem with the fumarate because it was hygroscopic, had all the typical salt-based issues. And there was a discussion, should we go further or not? And when you think on those lines, we want much more or a little more data to make that decision. And that's where we use the mini transfer model. So when you use the mini transfer model, we did it first for the parent compound. And as you can see, we see here again a supersaturation. And the supersaturation was being maintained for a significantly long period of time. The best part is when we did the fumarate salt and we did a transfer with the fumarate salt, it behaved exactly like the parent. The reason being, the drug had to move from the stomach environment into the intestinal environment. And that showed, if you look, the two profiles are so different from the individual profiles. And that way,
I'm sorry to interrupt you, but there has been some problem with the presentation. As in the YouTube stream, people are not able to see it. So if you could stop presenting and present it again, probably they'll be able to see. Yeah.
I can try that. Just a second. Sorry about that.
No, it's okay.
Okay.
You stop and then again start.
And then I have now stopped presenting and I am starting again. Can you all...
And then share.
Yes, I have. And now can you see my slides?
No. Yeah.
Yeah.
Yes?
Yes.
It's
Okay.
visible? Okay,
Yes.
perfect.
Yes. But make
Thank you.
slides. Okay, now. Good.
Yeah, okay. So sorry about the technical issue. So coming back to the point. So as I mentioned, the fumarate salt behaved quite similar to the parent. So which being said, we could very easily then think that if the behavior is similar, why not go with the parent for further development? And the stability issue of the fumarate salt no longer was an issue anymore because the parent was the most stable form. So those are the findings that you can do when you have this data early on. Or it could have happened that because of stability issues, you may reject the parent and you may reject the fumarate also, and the molecule may not go further. And that's exactly that I was trying to address when I said potential molecules can be then screened out if we don't have the right data for the decision making. Going further, this is with regards to formulation design. Probably you would have seen these kind of graphs very often. But we know that when the drug moves from the stomach into the intestine, there is a tendency for precipitation. When it precipitates very fast, usually amount of drug in solution is low, so the solubility is also lower, bioavailability is lower. But when we talk about formulation designing, we can add precipitation inhibitors like polymers into the formulation, which can delay the precipitation rate. This is called the spring and the parachute effect. So if you have a drug which is showing poor solubility characteristics, you can even check out whether the drug solubility or precipitation rate can be delayed by adding such inhibitors, and if you can enhance the solubility or the dissolution profiles. This is one example. If you see this light blue line, this represents the dissolution in a normal passive medium, no transfer. But this is the profile with the transfer. The green one represents with HPMC and the violet one represents without HPMC. If you look at the two, there is no much difference. So, which is similar for both the cases. And hence we came to a decision, okay, excipients do not help here to enhance the solubility. It remains the same. So let's avoid, we don't have to think in that direction. We can, exclude precipitation inhibitors and think about some other way of, bioavailability enhancement to see if we want to push the solubility or the dissolution higher up. Now coming to the last case study, this is also a published report about albendazole. So here I said that you can use a transfer model for formulation comparison. So this was a study done by Johnson & Johnson in Belgium, where they created for albendazole a molecule with lipid as a lipid suspension, and they made it with cyclodextrin, a solution. If you can look at the two profiles, so these are the profiles from the transfer model, you see that we have a higher concentration for the cyclodextrin solution in comparison to the lipid suspension. So if you do a rank order, we can say that the cyclodextrin is the first preference and the second preference is the lipids. In order to see whether the behavior would be same, the same formulations were tested in rats. And as you can see, the area under the curve for the lipid was much lower than the cyclodextrin. So this is one example where we say that yes, the method works, and so you can use the transfer model for the screening or rank order of your formulation, and you do not have to go for animal studies, and you can exactly tell how your formulation behaves. So from these examples, it is now clear that we have an array of applications in the drug research and development phase with a transfer model, and it can help us not only to troubleshoot, but also generate adequate data in order to get good information, good input data, so that we can put it into the respective models and understand the molecule better. That being said, the question here is this the one and all, or is this the final solution to all our dissolution problems? And the answer is no, it is not. So why is it not an answer? If you see, the model could mimic gastric emptying, it could mimic the conditions in the GI tract, but it couldn't mimic permeability. And you all know permeability plays a significant role in creating a sink condition for a molecule's dissolution. That means the data that I am generating here is a worst case scenario. It can happen that the molecule may do better than what I'm seeing in my dissolution models. So we need to keep that in mind, that the permeability aspect, I'm talking more about the passive permeability and not the other parts, because this happens to be the main permeability route that we are talking about. So that particular part of the model is not available. So then the question is, what if we add this model into the existing model? Then the question comes, how complex can you make your model? Because remember, we are comparing a complex human body to a vessel with a stirrer. It is not one-to-one. We cannot mimic it exactly the same. There, I will show you a next slide where they have tried to create various parts of the human gastrointestinal tract, like the tin model, where it behaves with exchanges. But then there it is so complex that sometimes you lose out to understand where exactly is the problem with my molecule. Is it the molecule or is it my method? So as you can see in this slide, these are the various models that has been published lately in the last 2 decades. This is very commonly used by a Swedish university to understand biopharmaceutical permeation. So it is very similar to the transfer, but they have another donor and an acceptor compartment which mimics permeation. This is used in the University of Athens, where they have a reservoir with pump in more facile and have a input and an output rate in order to mimic the sink condition. But for that, you should know what the permeability is. You should have some kind of human in vivo data. This is the tin model that I was saying. So they have a compartment for stomach, duodenum, jejunum, ileum. It looks very complicated. They even have a tiny TIM model, but then none of these is a solution for everything. Some drugs behave well in a model, some drugs do not behave well in the other model. So the question is, which one do you use? There is no one single solution for all. The point is, what is the data that I'm generating, and understand the data and the limitations of the model very clearly, and then accordingly use it in your prediction models. So to just sum up that last part, it is an advantageous model, but it has its own set of limitations because of the lack of permeability. So then the question is, do we stop? Is that where we are, or is that maximum where we can go to? To that, my personal opinion is human body is like a puzzle. And as Einstein says, the important thing is not to stop questioning. Keep being curious and always find out why and what this thing is going to do and how we can evolve into it. And if we continue to do so as scientists, I'm sure one day we will bridge all the gaps and we will be able to have better prediction and get out of the whole animal model aspect. So that's all from my side. And I think I can take around 2 to 3 questions before I log off completely. Thank you very much for your attention, and I'm open for all questions.
Participants, you may please post your questions in the chat box.
Okay, so till they type their question in the chat box.
There is a question, Priyanka.
Yeah, there is a question.
Okay.
Yeah. Anuradha Samant has asked, can we use these models to study pharmacokinetic herb-drug or drug-drug interactions?
Yes, you can do that. So at the end of the day, it's the molecule that you're going to put into it. Of course, when you're talking about herbal drugs, you need to have a very strong and good analytical technique because it's tough to get a very pure herbal formulation. But drug-drug interactions can be studied. For example, there has been studied to understand the effect of grapefruit juice on the absorption of certain drugs. And then what was done is they took in the intestinal medium, the facile media with some grapefruit juice in it. So you can just mix the two together in a fixed concentration and then see the dissolution profile and see how it affects dissolution.
Abha ma'am, you were asking something.
Dr. Anita,
Yeah.
Acha, can you tell me, during the pre-formulation studies, you are only going to use the drug, okay?
Yes,
So
yes.
how do you perform the dissolution rate studies of only the API?
The point is,
okay.
Yeah. The point is, in the beginning, it's before the formulation development starts, it is very important to first understand the dissolution characteristics of the drug itself. So, for example, as I showed you with the fumarate salt and with the parent, depending on the kind of dissolution setup that we use, the behavior can be totally different. So, before we add more complexity to the drug behavior, that is by adding excipients or any other stuff, or even the formulation steps, we should first understand how my drug molecule behaves. Once that is clear, then we can gradually keep on adding your excipients, the formulation, and then you will be able to understand clearly what is the effect of these additional excipients on the resolution behavior of the drug. It is very similar to the quality by design concept that FDA asks to do, because you know then in which space you are in and which parameter which I change can be critical. There could be some parameter that is not critical at all. And so it will not affect my drug drastically. But in order to understand, we have to go by a step-by-step gradual approach. And that's why the dissolution studies only on the pure drug is very important.
Anita ma'am,
Thank you.
can I request you to please stop sharing your screen so that we can take a picture?
Okay, so I need to see you presenting. Okay, I have stopped presenting. I hope so. Yeah.
Any more questions from the audience?
If nobody asks, then I will ask.
Please go ahead.
Go ahead, Abha.
Okay, so Anita, I think you must have worked on this mini transfer equipment. So
Yes.
tell me the working of this. I could not understand. Means from one container it is going to another container continuously.
Yes, so the point is this. So first let me just give you an insight into what happens in vivo, and then how we try to mimic it in the lab using the transfer model. So when we take a particular tablet, it is usually said that it remains in the stomach in a fasted state between 30 minutes to 45 minutes. Yeah. So the drug is exposed, so it has that time period to dissolve completely, depending on its solubility, or it would be in the form of a suspension. So partly dissolved, partly in particle form. Then the stomach starts pumping out these contents into the intestine gradually. Ideally, in the normal in vivo state, it's a first order. That means in the beginning, the first 15 minutes, it is usually a very gradual pumping that happens in our body. The speed increases, so it's an intermediate effect. And the last phase is called the housekeeper's wave. The housekeeper's wave is something like pushing everything. So you can imagine how your moms used to clean the floor where they put water and just push everything out from the floor. Very similar, the whole stomach pushes the contents into the intestine. Yeah. So when we go for an advanced transfer model, we try to mimic the same procedure based on the time using a programmable pump. But when you are in the preclinical phase, we do not have to go so much in detail, because the point is just to generate data in the beginning to see what is the behavior. We will fine tune it later in development when the amount of drug is more. So here we use a zero order kinetic, so only one rate, which is around 0.83 ml per minute, which is equivalent to, it takes around 30 minutes to empty the stomach contents into the intestine. So what we do is you put the drug into the stomach compartment and you allow it to be there for 30 minutes. You don't do anything, you just keep stirring. So it is mimicking that the drug is in the stomach. And then you start pumping the contents from the stomach into the intestine such that all the stomach content is emptied in 30 minutes. So we are mimicking here what is happening in the stomach and the intestine. And when the drug is coming into the intestinal compartment, you start taking samples after, for 3 hours. So at fixed intervals, very similar to how we take our dissolution study readings. And then we can see the supersaturation, the precipitation. In some case, it doesn't precipitate. In some case, it precipitates very fast. And if you have enough amount of drug to study, you can even take the residue of your drug in the intestinal compartment and do a DSC or a PXRD study to see if it's a different structure, the crystal structure. So sometimes, most of the times, we know that when drug precipitates, it precipitates in an amorphous form. And an amorphous drug have a higher solubility than a crystalline form, which is again advantageous for the bioavailability aspect. So there are a lot of things that can be studied, but given that, as I mentioned, the amount of drug is very low, we need to pick up the right data with the small amount of drug and try to capture such kind of the scenario of what can happen.
Thank you very much.
I hope I could.
Yes, you
answer.
could answer, but the tablet which was
Yeah.
in the gastric fluid, you will have to transfer that remaining portion of the tablet.
No,
Which
it generally disintegrates and then it moves and it will
Okay.
disintegrate, it will form a suspension and then it is pumped along with the fluids. So there is no filter between the gastrointestinal part and the intestinal part. It's just a tube which is going on pumping. So everything what is there will do that. Because the funny part is even the undissolved excipients can sometimes have a positive or a negative effect on your dissolution. For example, if you have an undissolved excipient, it can act as a seed for crystallization because you are supersaturated and the drug is waiting to burst out of the medium. And when you have an undissolved particle floating, I think all chemistry teachers know that where you ask us to scratch the base of the conical flask or put something, the same effect is happening in the intestine. And then you are having a really big problem with the bioavailability because there's less drug available for absorption.
Thank you. Very well answered.
Welcome.
There's one more question from the audience. Upu Mahesh has asked, are there any methods that
Yes.
use synthetic membranes to mimic the gut wall for estimating permeability?
Yes. So I think you are referring to pampa membranes. So there are several membranes that are available where the gut wall permeability is being mimicked. If your question was with respect to how to incorporate the so-called permeability factor within this dissolution medium, if you go into the website of Absorption Plus or Simulation Plus, sorry, not Absorption Plus, but Simulation Plus, they have the absorption systems also, where they have tried to have a sort of an additional container within the dissolution medium where the permeation can happen. Even Pion is trying such kind of setup in dissolution where such artificial membranes are used to do the permeability. It has worked very well for generics, but it is still under research for new chemical entities, because in generics we know how the drug behaves, but for new chemical entities it's still under research. It is one field of research that everybody is looking into, and yeah, if there are young minds like yours who can contribute to that, it will be a big breakthrough for the biopharmaceutics world.
Okay, last question for today. Surbhi has asked, to study more in this subject, what are the sources?
With regards to biopharmaceutics, I would refer to you initially into your textbooks. So I'm sure Abha ma'am's lecture is also very important for that. So if you see, biopharmaceutics is one subject, or I love this subject basically because it encompasses all the parts of pharmacy. Chemistry is important, pharmacology is important. So if you have a good basics there, that's really good. There are quite some books and textbooks available in biopharmaceutics by Shargil, which was one that I always used to refer. And then I would always recommend to go through review articles available in journals. These give you a very good overview on what are the new trends within biopharmaceutics. And if it is very oriented to dissolution per se, then I can recommend the European Journal of Pharmaceutical Sciences, where they very commonly publish such kind of papers. We come across such papers very commonly there. That is also one source that can be used.
Thank you, Anita ma'am. The question answer session is done.
Thank you very much. So it was indeed a pleasure to be part of this such a big event. I wish the organizers the very best. It was great to see my friends, my colleagues, their faces again. It's always a pleasure to connect, although virtually. Yeah, when I'm back home next time, I will surely be there. But thank you very much. Thanks for the audience. And if there is any questions, please feel free to reach out to Priyanka and she can forward it to me and I can always answer that whenever you have some. Yeah?
Yes, sure.
So thank you very much. Yes.
Anita ma'am, it has been a very nostalgic hour for me. I'm certain that the audience has
it was
also
more like a
benefited.
lecture, right?
Yeah. I'm certain that the audience has also benefited, just like we did as students, by your immense knowledge. You've not only recapped for us the basics of drug absorption, drug development stages, pre-formulation studies, physiological factors affecting absorption, but also emphasized upon the importance of early data in research and development. You introduced us to bio-relevant dissolution. Your lucid explanation of the need for mini dissolution models and their correlation with the larger version was very interesting. The applicability of these models were thought-provoking. Despite the limitations, the concept seems very promising. I'm sure we all will take home a plethora of knowledge. On behalf of MET Institute of Pharmacy, I would like to thank you for accepting our invitation despite of being in a different time zone and enlightening us with your talk. Thank you so much.
Thank you. Thank you very much. And thank you to everyone. And thank you, Kharkar sir and Abha ma'am, for the opportunity. Thank you very much.
Thank you, Anita.
Wishing you all a nice evening and a good session ahead and lots of success for your program.
Thank you. Okay.
Okay, bye-bye.
Bye.
So after the scientific lecture, we have planned for you all an exercise that will increase your concentration level and help you to focus better. So let's take a small break from academics to do this experiment. I now request Dr. Rashmi Srivastava to take over.
Thank you, Priyanka, and namaste to everybody. So there's a breather for breathing. Isn't it amazing? So we at MET always make sure that with the knowledge what we are giving to our students, along with that, we can give at least some amount of physical vigorness or rejuvenation along with the knowledge. So that's why we all have thought about this exercise. So we'll do few breathing exercises so that your concentration, your focus is increased. One more thing, in this COVID period, if you see this COVID is something who affects your lungs, and if we are doing these few breathing exercise every day, we'll be able to rejuvenate, we'll be able to increase our lung capacity. So I hope that you will utilize entire session by following my instructions. So be ready. We'll be doing few breathing exercises in which there are 3 rounds, and you'll feel fresh after this maybe 5 to 7 minutes exercise. First condition in this, that wherever you are sitting, sit comfortably. You can be sitting in a chair, on a wherever you are, just make yourself comfortable. Second condition for this is that you always sit in a position your spine is straight. So after fulfilling these 2 conditions, everybody just make yourself comfortable, make your spine straight. Now after doing this, what you are supposed to do is take 2 rounds of deep breathing. Inhale with me. I'll give you a count. 1, 2, 3, 4, 5. Now exhale. 1, 2, 3, 4, 5. Another round of deep breathing. 1, 2, 3, 4, 5. Exhale. 1, 2, 3, 4, 5. Everybody is able to follow, or should I be little slower? Okay, fine. So first exercise in this process is somebody, if there's any input, if you want to give, any faculty member, please just guide me. Okay, so first thing we are going to do is Surya Bhedan. What is Surya Bhedan? It is something wherein you will be inhaling using your right nostril, right one, and then exhale through your left nostril. We'll count for 5 inhale, and then 5 counts you will exhale from your left nostril. Can put your thumb on your right nostril, your ring finger on your left nostril, and these 2 fingers on your third eye. Inhale. 1, 2, 3, 4, 5. Exhale. 1, 2, 3, 4, 5. Once again, inhale from your right nostril. 1, 2, 3, 4, 5. Now exhale through your left nostril. 1, 2, 3, 4, 5. This is third and last round for Surya Bhedan. 1, 2, 3, 4, 5. Exhale. 1, 2, 3, 4, 5. So this is first exercise. Surya Bhedan is something which is related to heat. So if you want some kind of, because little amount of aggression is needed in few places. So if you want that kind of heat energy, you can do Surya Bhedan. Next exercise is Chandra Bhedan. Now Chandra Bhedan, what you're supposed to do, the way you've done Surya Bhedan, you will be inhaling through your left nostril for 5 counts and then exhaling through your left nostril only. So here you are not using both the nostrils, just left nostril is being used. So first inhale through your left nostril. One, 2, 3, 4, 5. Exhale. 1, 2, 3, 4, 5. Once again, inhale. 1, 2, 3, 4, 5. Exhale. 1, 2, 3, 4, 5. Third and last round for Chandra Bhedan. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. So we have completed 2 breathing exercises. One was Surya Bhedan, which is for generating heat in your body. So that can be done during winters. More rounds can be done. And second one is Chandra Bhedan, wherein you can do cooling, can get coolness if you are suffering from acidity, anger, heat. If you want to reduce those kind of things in your body, you can do Chandra Bhedan always. In order to balance these 2 nadis, because we have different nadis in our body, we do Anulom Vilom. Once again, we'll do 3 rounds of Anulom Vilom. That will be last breathing exercise. So will not take much from you. So what we do in case of Anulom Vilom, we inhale through right nostril and will exhale through left nostril. And then next cycle will be inhalation through left and exhalation through right. I repeat once again. Inhale through right nostril, exhale through left, and then inhale through left nostril and exhale through right nostrils. This will complete your 1 Anulom Vilom. We'll be doing 3 such rounds. Okay, can start with me. 1, 2, 3, 4, 5. Exhale. 1, 2, 3, 4, 5. Inhale. 1, 2, 3, 4, 5. Now exhale through right. 1, 2, 3, 4, 5. Once again, inhale through your right nostril. 1, 2, 3, 4, 5. Exhale. 1, 2, 3, 4, 5. Inhale through left nostril once again. 1, 2, 3, 4, 5. Exhale through right. 1, 2, 3, 4, 5. Last round for Anulom Vilom. 1, 2, 3, 4, 5. Exhale. 1, 2, 3, 4, 5. Inhale through left nostril. 1, 2, 3, 4, 5. Exhale. 1, 2, 3, 4, 5. Thank you so much. This finishes our 3 rounds of Anilom Vilom as well. If you want, you can once again 2 sets of deep breathing wherein you can involve your abdominal breathing. And while exhaling, you can always throw out negative things from your personality. So you can inhale confidence, whatever you want to inhale, positivity. And while exhaling, you can always throw out whatever is negative in your personality. So whatever you want, you can exhale out. So inhale positivity with me once. Confidence, good vibes. Anything you want to, you can inhale. Now keep on exhaling negativity. Tiredness, whatever you feel negative in your body, you can throw it out with that exhalation. Once again, inhale positivity. And exhale whatever you feel is negative in your personality now. Thank you so much. This finishes my breathing session. Hope you must have enjoyed and must be feeling rejuvenated. If yes, please do it daily so that you can beat whatever is there, that current scenario of affecting disease which affects your lungs, so that your lung capacity is always increased, your oxygen levels are always high. Thank you very much. Over to you, Vrushali ma'am.
Okay. A very good evening once again. I'm sure all of you are energized by the breathing exercises that Rashmi ma'am took us through, and we are all ready for our next session. Just a small request before we start to all the participants. Those who have joined us on Google Meet, henceforth, please join in by clicking the join in link and not on the present now link. Because once you do it with present now, it disrupts the complete flow of the presentation for the speaker as well as the listeners. Now coming to the next session. It gives me immense pleasure to introduce our next speaker for today, Dr. Prashant Kharkar. So Dr. Kharkar is currently working as a professor of medicinal chemistry at Institute of Chemical Technology. He is a drug discovery scientist with rich experience and expertise in medicinal chemistry, computer-aided molecular design, chem informatics, biomedical sciences, with more than 15 years of research experience, all industrial, postdoctoral, and academic. This research experience comes from his previous professional affiliations from SV Chems, NMIMS University, Global Discovery, Piramal Life Sciences, to his postdoctoral research fellowship at Department of Pharmaceutical Sciences, Wayne State University, Detroit. He is also currently working as a consultant to various companies to increase the institute and industry interaction. His expertise lies in computational and experimental drug repurposing. He has recently completed preclinical development of cancer stemness inhibitors lead molecule. He is also a registered patent agent and presently an inventor on 7 national and international patent applications. He also has recently filed a second use patent for COVID-19. He has guided 5 PhD students and more than 25 PG students, and so far has published more than 60 research papers in reputed international journals. He has also presented more than 40 abstracts in conferences of international and national repute. He is a recipient of grants of over INR 2 crores from various national funding agencies as well as the industry. He is also a proud recipient of many awards and grants from various government funding agencies and industries. To name a few, he is a recipient of Indian National Science Academy Award for International Exchange in 2015. And more recent, he is a recipient of the prestigious Newton Bhabha Links Workshop Award 2019. The theme being tuberculosis and HIV co-infection. He is also a reviewer of prestigious peer-reviewed international journals. And so with this very brief introduction, I request Dr. Kharkar to please take over and start with his presentation on drug-like filters in medicinal chemistry and drug discovery. Over to you, sir.
Okay, thank you so much, Dr. Vrushali. It's indeed a pleasure to see you after so long. Not so long, probably a year or so. Yeah, thank you so much for the kind introduction. I will, yeah, perfect. Now my screen is visible.
Yes.
Okay, so I will start with the presentation. Just a minute. Okay. Thank you so much to Dr. Rashmi for the to start with for rejuvenating everyone with the breathing exercises. It was indeed a pleasure to be part of one. And in fact, the theme of my talk is more or less similar. Taking out or leaving or getting rid of the negative things. So that's exactly what the theme is all about: drug-like filters in medicinal chemistry and drug discovery. Now, you may say that. What is it, why two things like medicinal chemistry is part of drug discovery? But as the story unfolds, you will understand what I mean by the use of drug-like and lead-like filters in medicinal chemistry and drug discovery. So with this, I will start with my presentation. The first part definitely is acknowledgements. I thank the organizers of the webinar and the scientific community to be, for everything, for that matter, for inviting me, to the organizers for inviting me for this talk. And I was told that majority of the students or majority of the participants are students from 3rd and 4th year. So that's exactly why I have made my presentation to suit their understanding and their level in terms of what they know or their knowledge. So some of the teachers, some of the chemistry teachers, or for that matter, most of the teachers may find, the matter is like it's probably already known to them. But in a way, I will add in my experience and expertise, whatever I have learned all these years since my ICT days back in 1998. And then over a period of time, I've worked in industry, I've worked in academia for postdoc. So a lot of interesting stuff I have got in terms of what I know and what I use in daily practices. So before I start, the first thing, the disclaimer: the views, ideas, and opinions expressed in this webinar are solely of mine. There may be some conflicts. Some people may feel there is a conflict, but don't worry too much. It's just my view or my opinion, and we have we agree to disagree in research all the time. So don't worry about it, as in don't mind. Okay. So before I start talking about drug-like filters and their usefulness in medicinal chemistry and drug discovery per se, I will just take you through the art and science of drug discovery. Don't worry, I'm not going to show you those interesting pictures where you have got all the different stages of drug discovery and how a drug is developed. So probably you've heard it many times, and I'm not going to repeat the same thing and save you some effort of not going through the same stuff again. Rather, I would tell you the real picture, realistic view, or I would say what exactly what happens in drug discovery. Like I was fortunate to be part of drug discovery companies in India, as well as the multinational drug discovery companies. And of course, I was in US for 3 years for my postdoctoral research. So in a way, I've met people who really were big shots in a way, and whatever I could learn from them, I'm just trying to present throughout this seminar, not just this slide. So when it comes to drug discovery, it's awfully difficult. I'm sure you have heard about this from many people, many scientists or researchers. Drug discovery is awfully difficult, more so in the current situation, as in I'm not talking about COVID, I'm talking about in general. In the 21st century, for that matter, right from the early 2000, after the human genome sequence, human genome was completely sequenced, and we knew we had more than additional 1500 odd targets which we can use for developing newer drugs. As in, the productivity should have gone up, but it's actually downward trend, whereas the spending has increased way too much, just like you see in any app, how many number of COVID patients are increasing. Just like that, the drug discovery expenses have gone up. That's exactly why the next statement says drug discovery is expensive. You will hear or you will read about many more different estimates as to how much money it takes to bring a new medicine to the market. The current estimate is around average estimate is around $2.6 billion US dollars. Now that's a lot of money, and moreover, drug discovery is a gamble in the sense that, like, you put in your money, but you don't know whether you will win. If you win, you get in multiples, but if you don't, then your money is gone. Now, just to tell you a simple fact, that only 5% of all the drugs which are introduced in the market, they actually fetch, or they actually get the return on investment. Otherwise, all others, 95% of them, they can't even recover the money which has gone in their development, right? So it's awfully pessimistic view, but then that's the reality. Drug discovery is time consuming. It takes anywhere from few years to, like, say 9-10 years to some drugs they have taken like 18 years, 20 years. Don't worry about the COVID drugs. They will take forever. Like, so after the first outbreak of COVID-19 back in 2002, 2003, we still don't have drug for SARS-CoV. So leave aside SARS-CoV-2. We know a lot about drug repurposing, but that's a different story. All right, so drug discovery is time consuming, but for a scientist like me and many others who are attending this who are in the participants, drug discovery, they will agree to this very fact that drug discovery tests your intellect, your perseverance, your patience, and your analytical bent of mind. As in, you can't ever be sure that your drug will make it to the market, right? Some of you might be working in drug discovery, as in very early phases of hit discovery, lead discovery, or maybe if you're fortunate, lead optimization. But then you are like way too away from the drug being a reality or coming to the clinic. So it's despite all these pessimistic thoughts or pessimistic statements, drug discovery is still very exciting. As in, all you need is one drug. As in, I don't believe that a single person can discover a drug, but if you are part of some drug which actually becomes a reality, it's like you leave behind a legacy, as in your parents or your friends or whosoever, they will say, oh, this guy has discovered, or rather he contributed in discovering a drug. So that's a great thing. But yeah, it will take a lifetime to get a drug out in the clinic, right? And then of course the most important, or other last most important statement in this particular slide is there is no shortcut to a drug discovery. Yeah, so that's exactly what I said. Drugs for COVID-19, God knows when they will come. Okay, just to amuse you with some fun facts. I mean, they're very controversial. Well, they're not controversial, but you may find them kind of controversial, but they are not. As in, these are just fun facts. The biggest pharmaceutical companies in the world spend most of their money on marketing, not on drug research or developing new therapies. Yeah, well, you may say that I just told you it takes around $2.6 billion to develop a drug, to bring a new medicine to the market. And then I'm making a statement that most of the money is gone on marketing. Well, indeed, it is true. If you look at some real good statistics on the expenditure in drug discovery and development, you will see that most of the money goes on clinical development. Around 61% of the money goes on clinical development, right from phase 1, 2, 3, 4, and everything. And then 17% goes on drug discovery. Another 20% goes on preclinical. And then this the money on marketing, it's not even accounted for by in this particular thing. So you can do the maths, $2.6 billion, and how much they must be spending. But this is how it is. Only the USA and New Zealand in the entire world where pharmaceutical companies are allowed to create direct to consumer ads. Like if you go to US and look at, switch on the television, you will see many advertisements for Alzheimer's patients, for antibiotics, for a lot of things. You don't really see those in other parts of the world because that's not allowed. I mean, you have seen the story with the Patanjali medicine for COVID-19, right? But we are not here to discuss that. The next fun fact is pharmaceutical companies generate an estimated $300 billion in profit a year. So again, a controversial statement or contradictory statement. We are saying that they don't, take out the money, as in the return on investment, but here they are making the profit. So it's all the whole picture is very complicated. And if you read more about the way drugs are charged, or rather, how the things in US work, the #1 pharmaceutical market in the world. So the insurance companies, they are very strong in the Western world, say particularly in US, and the whole picture is very different than what we have in our country, that is India. So we should be thankful to our government and everybody for that matter. The medicines are so very cheap and available to us. Sometimes we get it for free, okay, but we don't realize the importance. Anyway, the next part is now, before getting into the drug-like filters, so the initial discussion is to make you aware of what is the importance, like why drugs fail in the market, in the clinic, meaning you might be reading about many cases or many case studies wherein a particular drug is withdrawn from the market for this reason. So the company spends so much money, and then eventually the drug is withdrawn from the market, say maybe after 2 years, 10 years, or maybe God knows what. And then on one side we have got drugs like aspirin, which was introduced back in 1896 or early 1900s. We still use aspirin, right? So it's all every drug is different. Every drug has its own lifetime. Some drugs are like perpetual, whereas some drugs are short-lived. So if we understand why drugs fail in the clinic, that's one thing. So right, as in in phase 4 clinical trials after they're introduced in the market. And another thing is, I'm sure you have heard about the statistics, like for every drug which comes out in the clinic, somebody has synthesized around 10,000 compounds, back in the drug discovery phase, and then some of them they got into preclinical studies, and then from there 100 of them entered in phase 1 studies, maybe 10 in phase 2, maybe 4 in phase 3 and then only 1 comes out, right? So you see the attrition rate. So whatever gets left behind, say in phase 1, phase 2 and phase 3, I mean those are expensive things for a company, because at times a company is putting the investors' money on developing the drugs, and if the drug doesn't do well, then obviously the company share will go down. They'll lose a lot of money in terms of the market capital and stuff like that. So it's very apt for us to understand why drugs fail, as in if it is such a lengthy process, why do when they fail, right? So that's exactly why we will just take a quick look at 1 or 2 cases why drugs are withdrawn from the clinic. So this one is a drug named lorcaserin, which was introduced by a Japanese company, AC Pharma. Now the structure is shown here. It's a very tiny structure, simple structure. It was withdrawn from the clinic in 2020. I think there was a study conducted by FDA. This drug is already in the market, probably for 4 or 5 years it was in the market, and in a big phase 4 clinical study, the FDA involving around 12,000 patients, FDA found out that it indeed led to increased risk of cancer in patients who were using this. So then you may wonder, why was this like? Why did patients use this drug for, or what did patients use this drug for? So this was basically used for weight loss. I mean, it's a big market. And lockdown probably has created more opportunities for such medications where you are interested in losing weight. So basically, this drug acts by by agonism at selective agonism at 5-HT2C receptor. It's a CNS acting drug, and eventually, it is used in obese individuals. But it was withdrawn. FDA issued a regulatory notice or something to the company to voluntarily withdraw this particular drug from the market, and the company did follow that, right? So my question to my young friends, my student friends, look at the structure carefully, and I want you to maybe take a note or something. Anything appears troublesome to you in the structure, right? You can say anything. Probably the benzene ring, or the aromatic halide, or maybe the secondary amine, or maybe the benzylic methyl group, anything. I mean, it's a tiny drug, right? So just hold on to that thought. Maybe you can think over what could be wrong with this drug, right? So this drug, in a way, completed all the preclinical, clinical development successfully. It came to the market was in the market for at least 5-6 years, and one fine day they just say that, oh, okay, it's time, your time's up, you get out of the market. So you can imagine how difficult for the company it would be, and more so the patients who in a way kind of develop the cancer by use of this drug, how situation could be for them, right? So it's very kind of troublesome for everyone. This is another drug by the company Boehringer Ingelheim from Germany, clobuterol. Now this particular drug, if you see again, before I conclude this slide, I will have the same question. Look at the structure carefully and what is wrong with this particular structure. So this drug was withdrawn in the clinic in 2007. It was again it was in the market for quite some years, if less than probably 10 years or so. The reason for its withdrawal was potential to prolong the QT interval. Now QT interval, so I'm talking about cardiotoxicity when you have prolonged QT interval. So basically there is this risk of arrhythmia called Torsades de Pointes, which in a way increases the QT prolongation, and the heart rhythm goes haywire, and there's a potential risk to the patient could die of the cardiac complications, because this drug has had the potential to prolong the QT interval, right? So again, the mechanism of action, it was actually used as a cough suppressant. So if you look at the structure carefully, you would figure out that this has got structural similarity to, say, sympathomimetic drugs or sympatholytic drugs. So you've got a tertiary amine, you've got a hydroxy group, and then you've got a benzene ring and stuff like that, right? So again, the same question, anything appears troublesome to you in the future? So if you are vigilant enough, you will see that, oh, okay, I got it. You've got an aromatic halide in the previous drug, you've got an aromatic halide here. So, but again, when I selected these cases yesterday, I did not want to be biased because I know how things. I have seen hundreds of such cases, so I can be very biased in selecting these drugs, and I can give you a wrong picture. But don't you worry about it. If you just type in Google or rather Wikipedia and search for drugs withdrawn from the market, it will give you a huge list of drugs which were withdrawn probably over like last 3 or 4 decades. You can see for yourself. Now, not that every drug will have aromatic halide, but as a drug discovery researchers, I would definitely hate to see an aromatic halide in any drug for that matter. You can probably check all of my papers, and hardly you will find out any compound having an aromatic halide. I don't deny that it's not there, but at times the student was persuasive enough to kind of convince me to include that drug in the paper, or rather that molecule in the paper. But I would do it with with no involvement of mine, as in I would be hesitant to you work with such drugs. Okay, so having said that, so this was the first two cases was drugs which were withdrawn from the market. So similarly, there are a lot of phase 3 failures due to lack of efficacy. So if you look at again the statistic why drugs fail in clinic, in the sense before they're introduced in the market or in the clinic. So clinical development, why drugs fail in the clinical development, and phase 3 failures are the costliest, because if you look at the number of patients which are included in phase 3, so depending on the indication, depending on the disease, you could have any number from 600 to 10,000, right? And you have to pay these patients, you have to monitor the therapy, the outcome. So it's a huge cost. You have to pay to the physicians, and you have to pay to the site, and the regulators and stuff like that. So if you have a phase 3 failure, that's really I would say that's a shame, I would say. But as in not shame in a bad sense, but as in, like I said, it's a gamble. Anything can happen. So the companies know it very well. But then you may my student friends may ask, what were they doing? Why would they just observe lack of efficacy in phase 3 and not earlier studies? Again, there are several reasons for this, but one of the reasons being the number of patients which are included in phase 1 and phase 2, that's quite low, and at times such things are not picked up. And whenever you have the statistical analysis done for such studies, so at times you may see that there is no statistical difference between the placebo and the drug, the treatment, and that's where you say that there's lack of efficacy. Because if the FDA is not convinced that the drug indeed has more beneficial effects than the side effects or adverse effects it causes, they're not going to grant you any permission or marketing authorization approval. Okay, so again, two more studies. If you again go to US FDA website, there are like lots of studies where drugs failed due to lack of efficacy. So efficacy is as in there is no effect, statistically significant effect seen in the patients for whatsoever indication the drug is prescribed for. So the first case is Iniparib. It is from Sanofi, the French company. Again, you may say, some of my chemistry friends who are really good at chemistry and the faculty say that I am doing this on purpose, that there is a halide and then there is a nitro group. So we know it from in our wildest dreams also that this drug is likely to be going to be bad. But believe me, this drug indeed showed very good activity right in the animal models and even in the phase 1 and phase 2. But then the development halted due to disappointing phase 3 results in 2030. Again, this drug was, they were developing it for triple negative breast cancer, for which there is currently no treatment. There is some palliative treatment, but no definitive treatment for sure. 50% of the patients, they don't really respond to anything, right? No matter what you try. So lack of efficacy in triple negative breast cancer patients, female patients. The mechanism of action. So originally, it was thought that it would act as a covalent modifier of a protein, a target called PARP1 or polyADP ribose polymerase 1. But this guy just did not work when it where it was supposed to work, that is in phase 3, and very disappointingly, the company had to withdraw this from the clinical development, right? So again, the same question: anything appears troublesome to you in the structure? And of course, I have already told you, well, benzene ring, it's there like in almost every drug. So you've got an aromatic halide, you've got a nitro compound. So what's the fun in that? As in, but okay, this is another one, and I made sure that there is no halogen, as in the bad halogens, kind of chlorine, bromine, and iodine. But you have got fluorine. Fluorine is considered not as bad, and it is in the form of a CF3 group. But nonetheless, it is still bad, and this is the drug torcetrapib from Pfizer, USA. The development halted due to excessive all-cause mortality in the treatment group receiving a combination of atorvastatin and this particular drug, that is torcetrapib, back in 2006. So you have guessed it, this drug basically is an anti-hyperlipidemic drug. It's just that it doesn't act on HMG-CoA reductase, at which atorvastatin acts. Like atorvastatin inhibits HMG-CoA reductase. This drug acts a similar enzyme in the similar lipid biosynthesis pathway. That pathway is cholesterol ester transfer protein, CETP. So this drug inhibits CETP. CETP is responsible for transferring the cholesterol group from HDL, which is the good cholesterol, to LDL and VLDL. So this drug stops doing that. So in a way, the ultimate effect is you get elevated levels of HDL, and you get all good things, because there are very few drugs which increase HDL. Atorvastatin does that to a small extent, and by the way, atorvastatin is a blockbuster drug, right? But then this guy again gave up in phase 3, and they had to withdraw it from the clinical development, that is for phase 3 back in 2006. Again, now the same question. Now you may say that this guy is actually boring, but believe me, you will eventually learn what is so bad with the structure. As in, I always believed that everything is in the structure. I mean, it's the structure, it's the drug molecule which is going to go in the body, going to go, in the earlier very excellent lecture, you've seen the biopharmaceutical aspects of drug development. And then, of course, Dr. Nair also mentioned that the NCE that always, or NCEs always present new problems, because not many things are known about NCEs as compared to the drugs which are already made it to the market. So the thing is, one has to be very vigilant. So rather than these drugs either failing in phase 3, phase 2, and even withdrawn from the market, if we can do something in some way to identify these problematic candidates right in the drug discovery phase, so we'll save all the trouble in developing the drug till clinical development, and then eventually avoid such things. And before before 1996 or before 2000, the number of drugs which otherwise would fail because of poor pharmacokinetics was very, very high. It was more than 20%. Right, the number was very high, but then people realized, like the scientific community or the pharmaceutical industry realized that this is a very costly affair, that the drug is failing in late clinical development. So let us do something in unique way, that these problematic candidates are identified well ahead of time, that is in the preclinical development. And that's how early ADMET studies came into existence, high throughput screening, and then simultaneously combinatorial chemistry or combinatorial library synthesis concepts also developed. And eventually everyone understood that the importance of early ADMET studies, right? So eventually in like post 2000, the number of failures due to poor pharmacokinetics were brought down below 10%. Now that's a great, I would say, a great success. The early ADMET studies has offered, because the companies would save like millions of dollars, or probably billions of dollars in development, right? So like I said, at times the reasons why a drug is failing could be any and many, but most of the times it is some or the other thing related to the structure, right? And if you can pick that some or the other thing which can create problem eventually in later phases in clinical development, or even for that matter in animal studies, basically you eventually would avoid all the trouble, right? But then at times you feel that some of these things are not even closely related to drug structure. So the such reasons could be poor clinical trial design, poor monitoring of patients, poor therapeutic dosing, and God knows there are like hundreds of reasons why drugs eventually fail. But then we'll focus on the reasons why drugs fail in the in the clinical development, as well as in the after they enter in the clinic. And that's where this particular paper which appeared in 2015, what they did was very simple studies. And I must tell you, all the students who are listening to this, and maybe whenever you listen to the recording, you should read to this, go through this paper. It's from Nature Reviews Drug Discovery. It says what the authors have done is they have analyzed, or they have rather the combined data of on the attrition of drug candidates from 4 major pharmaceutical companies: AstraZeneca, Eli Lilly and Company, GlaxoSmithKline, and Pfizer. And then they compiled the observations, like why drugs underwent attrition in different phases, what were the causes, what were the whatsoever was the reason. But then if you read out the highlighted part, the analysis reaffirms that control of physicochemical properties during compound optimization is beneficial in identifying compounds of candidate drug quality, and indicates, now this is important, indicates for the first time a link between the physicochemical properties of compounds and clinical failure due to safety issues. Now, whether the compound fails because of lack of efficacy or due to safety issues, doesn't matter. As in the compound failed, it failed, right? And it has something to do with its structure, right? So those who work, particularly the chemists, those who work in drug discovery, they would agree to me that, I mean, if when I read this paper for the first time, this was no surprise to me, because we have seen similar cases. Eventually the drug structure had to do something with it being failed at some stage in the clinical development. Not that I was part of many clinical development programs, but whatever I have seen, say in Nycomed, a German company, and currently I'm a consultant to Godavari biorefineries, and in fact we have come up with a drug which will soon enter in phase 1 trials. And I have been associated with them for last 7 years, and right from the day one, when I joined, so we actually they had some natural product lead, and eventually we modified it. And I proudly say that my student in NMIMS, he was the first one to to synthesize this drug for the first time in our lab, like in the lab in NMIMS, and eventually the same drug actually sailed through. So then again, that's a very interesting story. Maybe some other time I will share. The point is, what I'm trying to tell you is, the chemist who is working in drug discovery, a medicinal chemist, I would say, would know something is not right with the structure. This is like girls knowing just by looking at a boy. The girls will have this sixth sense, who will understand this guy is not right. The reason may not be known, but then the guy is not right. So it's that sixth sense that a chemist otherwise would know something is not good with these compounds, and it is going to create a problem, right? So that's exactly you should read this paper, an amazing paper, and whatever they have come out with. I'm not going to present what they have come out with, because that's not you can read for sure, because the main purpose of this lecture is to generate interest. And believe me, my third year and fourth year B.Pharm friends and others, master students, my own students, you would definitely be interested in the subject. And the more you read, the more you know, and then eventually you can garnish the interest in the subject. Okay, so now, so this paper basically showed the link between physicochemical property and the compound success, either on the efficacy side or on the safety side. And that's where the physicochemical properties come into picture, and they are somehow or rather intricately related to drug action. Now, some of my master students whom I'm teaching biopharmaceutics and pharmacokinetics, so we have had this discussion many times. So for them, this would probably be kind of repetition, but just to for the benefit of everyone, when we talk about physicochemical properties, right, so and drug action, so in fact Dr. Nair has given you all the the great overview of what exactly happens from the biopharmaceutics point of view, how drugs from the formulation they get into solution, then they get into the bloodstream, depending on the route of administration, and once inside the blood, it goes to liver, from liver to heart, heart to lung, lung to heart, and again from there it goes to every single part of the body, right? So unless it's a targeted drug delivery, unless it's a different drug delivery, otherwise most of these properties which are listed here are true for all the drugs which essentially are administered by passive diffusion and given by oral route, right? Because that's what majority of the drugs are administered by. So the first thing is aqueous solubility. So definitely, unless the drug goes in solution, it's not really going to go anywhere. I mean, there are some drugs. The other day I was listening to a webinar from Dr. Michael Walker. So he said about one particular formulation. So the drug is given intravenously, and you can actually see the drug being precipitated the moment it goes in the vein. And over a period of time, it actually goes, it dissolves. But then we have those cases rare, probably one or two. So I'm talking about aqueous solubility. So unless the drug, irrespective of whichever route of administration you are using, unless the drug is solubilized, it's not going to go anywhere. So there are many cases of drugs in the development which failed because they did not have appropriate aqueous solubility, and none of the solubility enhancement parameters or approaches or strategies could enhance the aqueous solubility. So then the next one is C log P, that is calculated log P. Again, this is very important property. So the first the drug has to be in solution, and then once in solution, the drug has to cross, say, the intestinal barrier or the epithelial lining of the intestine, like human intestine, for an orally administered drug. And more so, the drug has to go through innumerable number of cell membranes before it reaches its target site or site of action. So C log P, calculated log P, is very important parameter. So unless you've got an optimum value of log P, the compound is not going to cross the hydrophobic interior of the membrane and get into the aqueous environment. So initially the compound is soluble in the aqueous environment. It crosses the highly lipophilic membrane, goes inside, and again it has to cross the basolateral side of the epithelial lining to get into the blood capillaries and then endothelial cells, and you have got all the different methods by which the drug is transferred, the transcellular route, paracellular route, and then of course active transport, but we're not talking about active transport. So when you say C log P, so we are solely dealing with the passive diffusion or drugs absorbed by passive diffusion. So C log P, C stands for calculated log P. If you experimentally determine it, it will be called as just log P. And you all know that it's a ratio, or it's a logarithm of the ratio of concentration of drug in the oil phase versus concentration of drug in the water phase, right? So you can actually eat your chemistry teacher's brains for for knowing more about this if you don't know. The next important parameter, or rather I would say more so more than C log P, I love log D, that is distribution coefficient. Again, C stands for calculated. So if you are predicting it using some software that essentially gives you the calculated value of the distribution coefficient. Now it has got the same meaning as log P or partition coefficient. It's just that it takes into account all the ionized as well as unionized forms of the drug, right? But in case of log P, you only talk about the unionized form of the drug, and that too at pH, at neutral pH, pH 7, or maybe you can say 7.4. But in case of log D, you can have different values: log D at pH 6.5, log D at pH, which is the duodenum pH, log D at pH 4, which is the pH on full stomach, log D at pH 8, which is the pH in later part of the intestine, right? So depending on as the ionization status of the drug changes because of the pH of the external environment, the value of log D will change, right? So the basic difference between log P and log D is log D takes into account the ionization behavior of the compound, whereas log P does not. Okay, the next one is TPSA, or which is also called as PSA, or polar surface area. TPSA stands for topological polar surface area. So basically, this is the surface area contributed by polar atoms in a molecule. When I say polar atom, so any atom which is different than carbon and hydrogen. So you're talking about if you say commonly used atoms in drugs, so you've got oxygen, nitrogen, some halogens, you've got sulfur, you've got phosphorus, you've got some drugs contain selenium, some drugs contain silicon, but some drugs contain boron, but then those numbers are relatively rare. So any heavy atom other than hydrogen, heavy atom is other than hydrogen, and a non-carbon atom which is contributing to the surface of the molecule, the polarity, because the atom is polar, that element is polar, so the surface area contributed by those atoms is called as topological polar surface area. So it's actually a fraction of total surface area to the surface area contributed by polar atom. So polar atom surface area divided by total surface area, that will give you into 100, that will give you the topological polar surface area, or it is just a fraction of the total surface area, right? Then number of rotatable bonds. This is another very interesting property. If you see some old drugs, if you are studying some drugs which are cardiovascular drugs, say antiarrhythmic drugs like verapamil, or maybe other drugs, calcium channel blocker, that's verapamil, which is also used as antihypertensive and for other indications. So many drugs which were discovered like ages ago. Another example could be haloperidol. So some of these drugs have got many rotatable bonds. So as the number of rotatable bonds increase, say goes beyond 8, essentially the metabolic liability of the compound increases, right? And that's where you would want to control the number of rotatable bonds right away, right from the drug discovery phase, because as the compound moves from, say, hit discovery to lead discovery to lead optimization, the compound becomes fatter and fatter and fatter. I will come back to that just in few moments, right? Then another important thing is number of aromatic rings, right? So you don't want to see too many aromatic rings in your molecule, because then obviously there will be other issues in the molecule which will pop up. Could be liver toxicity, could be anything, could be any toxicity because of the metabolism, higher metabolic issues or liability. Anything can happen, right? So number of aromatic rings is another important parameter. Number of acidic, basic, zwitterionic or neutral group. Again, if you remember pH partition hypothesis in medicinal chemistry 1, so which says that usually, like, most of the drugs are absorbed by passive diffusion, and it's only the neutral form of the molecule which gets to cross the cell membrane, right? And you have to cross like hundreds of cell membranes in the body. So the nature, because most of the drugs are either weak acids or weak bases, so eventually the number of acidic, basic, zwitterionic and neutral groups has something to do with the overall pharmacokinetics, and also with the its success in the clinical development phases. Another interesting feature is number of hydrogen bond donors, right? So hydrogen bond donor is, I'm sure you know the concept of hydrogen bond. So any group which contributes a hydrogen in the hydrogen bond formation is called as hydrogen bond donor. So the number of hydrogen bond donors are extremely important, because this number will eventually affect your aqueous solubility. This number will eventually affect your lipophilicity, even for that matter distribution coefficient. This number will eventually affect the present, or rather the TPSA. So everything is like interlinked, and depending on what context you're talking about, so you will find that one property has got a direct effect on the other property, maybe in a positive way, maybe in a negative way. Similarly, I've got hydrogen bond acceptors. So basically, again, it's the same concept. Any atom which accepts the hydrogen bond is called as an acceptor. You can read through more if you don't know what hydrogen bond acceptors are. molecular mass, of course. Now if you look at the drugs which are introduced in the market in last 10 years, so on an average the molecular weight is way beyond 500, so around 600 or so. So that's exactly what they keep saying, that all the easy drugs have been discovered and it's like the toughest one are being discovered, and these molecules are kind of fat molecules, so they're like, they come up with huge molecular mass, and that actually creates a lot of problems, including the higher lipophilicity problem. And then eventually this higher lipophilicity is seen in various toxicity issues, and even for that matter the efficacy issues. You never know, right? Then comes the other part now, which is actually a separate thing, reactive or toxicophore flags. Just the way I showed you some of those structures which were withdrawn from the market or which failed in phase 3 clinical trials. So the aromatic halide is a reactive or toxicophore flag. So in fact, in the beginning, like I said, I don't want to see a nitro group in any of my molecule. I don't want to see any halide in my molecule, be it an alkyl halide or be it an aromatic halide. Just that way, I would take care of that right in the beginning, that I will screen the library so that all the reactive or toxicity causing groups, toxicophores, are taken out or filtered out, right? So that's why, although I'm not really talking about reactive or toxicophore flags, but then some of these properties eventually give rise to the toxicophoric groups. So it's like ketone is in a way a toxicophore. I would not want a molecule made in my lab to contain a ketone group, right? Absolutely not. Then even I would not want to see any molecule which contains a thiophene ring, a furan ring. Absolutely not, right? So that's what. So these are like strict no-nos in drug discovery or drug design and drug discovery, for that matter, or in medicinal chemistry. So back to basics. Importance of physicochemical properties early on in medicinal chemistry. So like I told you about hit discovery, lead optimization. It goes through many different stages. So coming back to that, the discussion will not be complete if I don't show you the bad picture first and then take you to the good side, right? Because then you will know what bad can happen. So that's where this particular drug comes in. The name of the drug is sulfasalazine. It's actually a prodrug. I'll show you the drugs. Now you see, this is sulfasalazine, an amazing drug. Well, it's not amazing. I mean, it is amazing, but not from the drug discovery or medicinal chemist point of view. So you have got a phenolic hydroxyl group, a salicylic acid moiety, you've got a diazo group and double bond, and you've got a sulfonamide, and you've got a pyridine ring, right? So because this is a prodrug used as an intestinal, used for the treatment of intestinal inflammation, which is called as ulcerative colitis, and even for that matter, infections of the large intestine. I mean, with ulcerative colitis and Crohn's disease, you have got those infections because the intestinal lining is kind of disrupted or inflamed. So what this drug does is, when it goes to the large intestine, now because this is so very polar, you say you've got OH, COH, sulfonamide, and double bond N, and pyridine. So this drug does not get absorbed from the GI. It's given orally. It's the dose is huge, but then this is not absorbed at all from the intestine, any part of the intestine for that matter, even for the stomach. So what the intestinal bacteria, particularly the colonic bacteria in the large intestine, they will chew off the N double bond N linkage using the enzyme called azoreductase, and you get 5-amino-salicylic acid that is on the left hand side. It gets separated from the other parts. We have got NH2 on both the parts instead of N double bond N, what you see in here, sulfasalazine. So, on the right hand side, this drug is a sulfa drug called sulfapyridine. On the left hand side, it is a 5-amino-salicylic acid, which is an anti-inflammatory drug. Sulfapyridine is an antiseptic drug. So this drug gets separated into both active moieties. So it's a mutual prodrug of 5-amino-salicylic acid and the sulfapyridine, right? So if I were to look at this from medicinal chemistry point of view, I would be worried about O and COH. Man, this is so polar, and it's going to create a lot of issues in terms of absorption. Yeah. Then to add to the vows, you've got N double bond N, which is highly toxic group actually. Then you also have got another interesting group, that is sulfonamide. And when it comes to medicinal chemistry, most of the sulfonamides, they create problems in terms of solubility, right? So that's why sulfonamides, most of the sulfonamide containing new chemical entities, they're like rocks. They just don't dissolve in anything, right? So that's another issue. And of course, the pyridine ring can get N-oxidized, undergo N-oxidation and can lead to toxic liver toxic or hepatotoxic metabolites. So you can see so many things in here. As in, its use as a prodrug is fine, but in fact, this drug is also showing some promising effect in for the treatment of glioblastoma, which is grade 4 astrocytoma, the CNS cancer. But then, because it is such a polar thing, it doesn't get absorbed from the GIT. Leave aside its absorption from the like crossing the blood-brain barrier. And moreover, the acidic groups are prevented from entering, or acidic drugs are prevented from entering in the from the brain, right? And that's where one has to just a minute. So I'm so sorry. So what happens is, somebody found that this drug has got a potent activity in glioblastoma in vitro studies, but then how to make it more druggable, and that's where we were faced with this particular problem. I can't make this drug molecule any better. It's like more druggable, where it can have a nice absorption. It crosses blood-brain barrier, goes into the CNS, and does what it does. So can you suggest something? That's a question to you. But then, yeah. The next one is, and then it appeared now. So this was the sulfasalazine was a typical case. What possibly can go wrong with drugs in terms of optimizing the physicochemical properties, right? And then back in 1997, this particular publication, which featured Lipinski's rule of 5, appeared in the literature in advanced drug delivery reviews. Now you see it has got several authors, Lipinski, then this Franco guy, Beryl, and Paul, right? 1, 2, 3, 4. Of course, Lipinski is the first and the corresponding author, and like he was probably leading the team, and eventually, this thing got into his head. They analyzed more than 2,000 drugs, and they came up with this Lipinski's rule of 5 filter. But then this was the seminal discovery in terms of drug-like filters, and you can read through this paper again. I'm just going to mention what Lipinski's rule of 5 filter is. Probably you already know what it is, but then just to reiterate, related to the theme of the particular like today's presentation. And you see this picture. Oh well, this is not. This is you may say that this is Viru Sahasrabuddhe. Well, indeed he is. He is not Christopher Lipinski or anything, or he's not the twin brother of Christopher Lipinski. Now the point of showing this particular picture, if you remember his dialogue in this movie, Three Idiots, nobody remembers the second person, right? Nobody remembers this person who landed second on the moon. Everyone knows Neil Armstrong. So just the same thing I want to reiterate here. There were so many of them in this particular publication who came up with this rule of 5, but it's the Lipinski who got all the credit for, right? So just remember this in your in your mind. You have to be the best, but then don't stress too much. Try your best, and you probably will be the best. This was just the fun part. Don't worry too much about it. Okay, so coming back to Lipinski's rule of 5. So what did he do? He actually analyzed almost 2,000 drugs, all belonging to all categories, including the ones which were absorbed by through human intestinal absorption, and other drugs which did not, right? So he came up with this particular four rules actually. It's called rule of five. So there are no 5 rules, it's just 4 rules. But then it says that if your drug has these properties, then you are likely to have good human intestinal absorption and possibly good oral bioavailability, right? So it basically relates to human intestinal absorption, and bioavailability is just the next stage of it, not necessarily it translates all of it into the the bioavailability. Okay, so what he says is your drug is likely to do good if it has got number of hydrogen bond donors less than or equal to 5. If it has got number of hydrogen bond acceptors less than or equal to 10, if it has got molecular mass or molecular weight less than or equal to 500 Daltons, and if it has got C log P less than or equal to 5, right? So a set of 4 rules, but everything is in multiples of 5. That's why it's called as Lipinski's rule of 5 filter. So this appeared in the literature in 1997, and around the same time the other descriptors or like other filters also appeared. I'm just going to mention some of them. So what did it do actually? It actually revolutionized the way the whole process, people, that time the combinatorial library just started appearing in the literature. So if I build a library and if I apply to the library this set of rules, so it is possible that, like, some ideas came to my mind, I put it in the in computer gave me enumerated that, and I have got a set of like 10,000 or 1 lakh or 100,000 molecules, right? But then I can't make all those 100,000 molecules. So what I will do is, the first thing I will do is I'll filter out those which are not likely to be successful in terms of the pharmacokinetics, right? So I will filter out those molecules which do not follow Lipinski's rule of 5. But then you may say that what is the guarantee that this rule will be true all the time? Of course, there is no guarantee, and in fact, there are many exceptions to Lipinski's rule of 5. If you look into the literature, you will find that there are many exceptions to Lipinski's rule of 5. So again, just to test this particular thing, what we did was, just to show you, now this is the structure of paracetamol, and using a tool called mole inspiration, all you have to do is just provide the smiles to, smiles is a simplified molecular input line entry system, which is shown here, this particular part. So if you draw a smiles and ask this software to predict the properties, it will do so. So this is the proprietary log-P, MI log-P, mole inspiration log-P, which is in a way a C log-P value. For paracetamol, it is 0.68. Its topological polar surface area is 49.33. Then molecular weight is 151. Number of hydrogen bond acceptors, that is O and N3. Number of hydrogen bond donors, OH, NH2. And it also says number of N violations to Lipinski's rule of 5, that is 0. Right. So let us get to the next compound, or rather, I would say. Now that I have calculated all of these properties, what do I do with this information, right? Like I already told you, all I can do is just filter out those 10,000 or 100,000 molecules which the computer has generated, given like when I ask computer to put my ideas into practice and enumerate the structures. So I've got a virtual library of 100,000 molecules, and then I use Lipinski's rule of 5 filter, and probably it will weed out maybe 60,000 molecules, just like that these 60,000 molecules are not worth looking at. But again, what is the guarantee that this particular rule holds true for anything and everything which I ask computer to do so, right? So that's where let us apply this particular case to sulfasalazine, where we know that the compound is so dead that it doesn't get absorbed in the human intestine. And that's exactly what we find out using the same mole inspiration tool. All other parameters are fine. If you look at number of violations to Lipinski's rule of 5, 0. So what does that mean? So if I were to discard, if I were to accept this molecule, suppose I was a drug discovery researchers when this molecule was in development, and then I thought I will take this molecule to to the next level, because Lipinski's rule of 5 gave me very good results. But the molecule passed, and finally we found that, oh my god, this doesn't really get absorbed, doesn't cross the human intestine, right? So what I'm trying to tell you is it's not foolproof. In the sense, it can give you good guidance in terms of filtering out the libraries, but then at times it fails for known drugs. One example is in front of your eyes. And then similarly, if you look at paclitaxel, which is awfully big molecule, the Taxol molecule, the anti-cancer molecule, very complicated molecule. So if you look at this particular thing, the number of violations are 2 shown in red. So the number of hydrogen bond acceptors are more than 10. It's 15 here, and the molecular weight is 853, which is absolutely fine. So probably this drug did not follow Lipinski's rule of 5, and that's exactly what happens. As in, it is Lipinski's rule of 5 says that this drug will not be absorbed from the human intestine, and essentially that is the scenario, right? But then people have come up with liposomal formulations and stuff like that, so that's a different story. So what I'm trying to tell you is, so you have to understand the importance of these filters on case to case basis, right? So the best thing is, you don't have to depend only on one particular filter, just like Lipinski's rule of 5. Like when I was doing my master's, so that was back in 1998, so Lipinski's paper was just published, and we kind of okay, there's something like that, and we used whatever we could back then. And if you look at my PhD thesis, there are like many molecules synthesized, and most of them, they would follow Lipinski's rule of 5. But then, but after that, a lot of things appeared in the literature, including other filters, right? So as far as the utility of this particular rule of 5 filter is concerned, so again, coming back to sulfasalazine. So if I have to develop sulfasalazine, like develop a library from sulfasalazine, which essentially would be more drug-like, so all I can do is I can probably mask the polar functional groups, increase its lipophilicity, and then eventually take it to the next level. But then in doing so, if I'm saying that I have to mask the carboxylic acid group, so I'll probably make an ester here. In the phenol group, I'll probably make an ether. But then whatever I put as a part of ether or an ester, I can I can put anything. But then when I design a library, suppose if I use 50 molecules, 50 alkyl halides, which will form ether with the phenol, and maybe 50 alcohols, which will form an ester with the carboxylic acid group. So my library size would be 50 into 50, that is 2500. But then I will not be able to synthesize all of those, right? Because it will be expensive, and maybe most of them, they may not turn out to be as good as what I would want them to. So that's exactly where once I ask the computer to generate a library of 2500 molecules, and then I apply the Lipinski's rule of 5 filter, and maybe 250 molecules survive the filter, right? And that's where the utility of these filters lies in designing a virtual library and scanning through a rule of 5 filter. So, and then you can probably look at only those molecules which follow rule of 5. But then eventually, since many people found out that the rule of 5 filter, how many violations should be permitted, and that's where they came up with the number that only one violation to rule of 5 is permitted, meaning you can break the Lipinski's rule of 5, but only on one parameter. Only one parameter can have value other or outside of Lipinski's, what Lipinski has prescribed, right? So that one violation is permitted. Just like this movie Chak De India, this guy Shah Rukh Khan's friend tells him that ek galti to sabko maaf hoti hai. Very similar way, one violation is permitted in this case as well, right? So having said that, let's move on to the next part. Now we are talking, as in now we know, okay, these things could be useful, and essentially I can now make use of these filters for taking or weeding out molecules, just like Dr. Srivastava or Dr. Rashmi Srivastava said in the beginning, that you weed out all the negativity. So exactly the filter is doing this thing for me. It's getting rid of molecules which are likely to be useless, and so that way, in a way, I'm saving a lot of money and time and efforts to make molecules which eventually would turn out to be bad or negative, right, for that matter. So that's what the rule of 5, RO5, helped me in optimizing my designs. But then since I know how these things work, so then probably when I think of a design next time, I'll will be very cautious of the molecular weight should not go beyond 500. The number of hydrogen bond donors should not go beyond 5 or something. So in a way, that calculations will start in my mind, and over a period of time, you will evolve as a medicinal chemist if you are into medicinal chemistry, and eventually you will start talking to structures, right? That's exactly I mean. If I look at the structure, if you give me some structure, if I look at the structure, I would probably figure out something that the structure is going to be good, the structure is going to be bad, at least say for pharmacokinetics, or even for that matter, toxicity. I've been working in this area for a very, very long time. The predictive toxicology and predictive pharmacokinetics, mostly predictive toxicology. So in a way, my eyes will catch those things very fast if there is something wrong with the structure, and that's exactly what happened in our program in Godavari biorefineries. Right. So interesting thing. Okay. So then, how do big pharma companies work? Right. Now, this one is an example from my own example, my own work. When I was working with Nycomed Pharma, we had a library of around 900,000 compounds. So every big pharma company has their own library. And these libraries contain compounds which have been synthesized in many, many different programs over like last 20 years, 30 years. So whenever a new program is initiated, the first thing they do is they will screen out all the library molecules in high throughput screening assays, right? So that's exactly what we did. So in this 2012 publications, so we had a task of coming up with a number of molecules, around 50,000 molecules from our original collection of 500,000 compounds. So the first thing what we did was we like our original library was 900,000 compounds. So we filtered out using all the drug-like filters, and we came up with 500,000 compounds roughly. And then we used other filters, including the pharmacophoric filters. Now these are activity-based filters, which eventually gave us a total list of 55,000 compounds, which were in a way present in our library. And we used those, out of these 55,000 compounds, we used top 50,000 compounds and got those screened for enzyme inhibition in an enzyme inhibition assay in GVK Biosciences in Hyderabad. And then you can read through that publication, the story will unfold. So the point behind showing this particular slide is big pharma companies will have very, very big collections. So they will screen all through whenever a new assay or new target is new program is initiated. So in a way they will find out some hits. So when a particular hit is found out, which initially was synthesized for a very different program or maybe a related program, so the first thing they do is they will chop off the molecule. In the sense, they will weed out the part of the molecule which essentially does not contribute to the activity or interaction with the target under consideration, right? So that's how they will first prune it for the given activity, and then they will regrow it by so-called during lead discovery and lead optimization stages. So what exactly happens is the hit and lead molecule will undergo a large number of structural optimization cycles, and so they'll generate lots and lots of compounds. Everything is put back into the compound collection, right? So the KID, when I say KID, the initial molecule which was pruned and which is now ready for further structural optimization. So during lead discovery, lead optimization, it will again become fatter and fatter, meaning its size will increase because you have to optimize other physicochemical properties. First thing is for target selectivity or target potency, because that's the first goal. You have to have very potent compound. The physicochemical properties come afterwards, right? So having said that, so this compound, which is initially a lead or hit molecule, which is a smaller one, it will go big, and then that essentially means it becomes fatter and fatter, right? And that's where you have to balance these physicochemical properties, and that's where it gets kind of messy, right? And many times the scientists will have to decide, there are decision gates. Say out of say 10 different series, the guys in the lead optimization says that give us only 3. So then they have to select out of those 10 chemical series or different chemical or diverse chemical series, they have to suggest the best three, right? And that's where these filters actually come handy, and essentially they help in making things easier, or life of the scientist, medicinal chemistry or drug discovery scientist, easier. So, but then, like I told you, only one filter, like Lipinski's rule of 5 filter, was not appropriate, because there were many exceptions which became very good drugs, but they never followed Lipinski's rule of 5. And that's where the literature started flooding with many different drug-like filters. And I mean, this field is so vast that one of from the student community, you probably can, if you're smart enough, you probably can come up with your own rule, right? So the sky is the limit in this. It's just a matter of you giving a thought and proving to the scientific community that indeed whatever you are proposing is useful. And that's where many people came up with many different rules. But then I did not put all of those, because what I felt was the purpose of this lecture should be to instigate you in terms of exploring it further. So on the same lines, the next rule was Weber's rule, which was published I think in 1997 or so. So only 98 or probably 99. So what this guy says is only 2 parameters. He says that the number of rotatable bonds should be 10, and the PSA, polar surface area, should be 140 angstrom square. Now this was fantastic. So it has an additional thing apart from what Lipinski just said. Lipinski never mentioned about rotatable bond. Lipinski never mentioned about PSA. Now when I say PSA less than 140, so that will eventually put a break on how many polar atoms you have. So eventually when you say polar atoms, so basically these are the atoms which will either be hydrogen bond donors or acceptors, right? So you control PSA, you control hydrogen bond donor acceptor. Similarly, you control rotatable bonds, you control lipophilicity. So in a way, these two filters are related, but you are just trying to take a look at, so this is like looking at Niagara from the US side versus looking at Niagara from the Canada side. From Canada side, it looks much better. I haven't seen it, but those who have seen, they would agree, and I have heard from people. Okay, so the point is, it's the same rule, like it's the same molecule, same library you are looking at, but you're looking at from a different spectacle, different filter, right? And so if you look at sulfasalazine, again it passes. It's such a tricky molecule. TPSA just at the border 140. So in such cases, as a medicinal chemistry, we wouldn't bother much about it. If it is 160 or so, I would say okay, and I would probably not consider this molecule. But these borderline cases, we tend to include them because we don't want to miss out something interesting. Similarly, if you look at the number of rotatable bonds, now number of rotatable bonds is around 6. So it actually passes both of the rules in a way, right? Although it's in TPS, it's borderline. So you can see it doesn't get absorbed, then it actually passes through these filters. So again, I would say this rule is not perfect. No rule can be perfect, and that's where you have to apply multiple of these drug-like filters and come up with your, you can kind of prune your library. So another one, now here, this is the structure of a drug called etoposide, which is an anti-cancer drug, or topoisomerase inhibitor. So if you look at the TPSA, it's 160 angstrom square, 160.86, and the number of rotatable bonds are 5. So out of these two parameters, it fails in one. So in that case, I would probably not consider this in terms of if my NC behaves similar way, I would probably not touch that, because the TPSA is more than 140, and then there is no way that this molecule will be orally absorbed. So Weber's rule says that to be able to absorb orally, this molecule must have number of rotatable bonds less than or equal to 10, and PSA less than or equal to 140 angstrom square. Then another guy came up with Indian guy, so you should feel proud. This guy, I think he is in at University of Purdue, and I don't know if you have heard about this drug called Darunavir. I think it is used for HIV/AIDS. D. Arun is the D. Arun Ghosh or something. So it's the same guy apparently who was involved in Darunavir discovery. So see, he came up with, he's a very, very good guy actually. So if you get a chance to work with him at Purdue, you would be, you would really be lucky to work with him. Okay, so the point is what he said that, so he proposed 3 filter, like 4 different parameters. He said that molecular mass has to be between 160 to 480, which is very similar to what Lipinski said. He said that log P can be between minus 0.4. So when I say minus 0.4, that means the compound is highly hydrophilic because the log P is negative to 5.6. So a little beyond what Lipinski said. Now, this is another parameter. Atom count has to be 20 to 70. So when I say atom count, it has to be heavy atom, non-hydrogen atom, which includes carbon. And molar refractivity, which in a way is also an indicator of molecular lipophilicity or lipophilicity in general. So it says that it has to be between 40 to 130, right? So this is the publication, which this was in 1998. This is yeah, this is a different guy actually. That Darunavir guy is different. I'm so sorry. Okay, so this is the Ghosh filter. Although the spelling you may say that it's a different, but this is how they do say. Anyway, the point is the filter which was proposed, the Ghosh filter, which is kind of people use it. I also use it sometimes. So this is the original publication where this thing actually came out back in 1998 actually. So that's where he proposed a lot of different things, right? So what I'm trying to tell you is, so again, for sulfasalazine, I did use a different tool for predicting the properties. This is from a tool called SwissAdmi. I'll just show you the screenshot. So you can see here, molar refractivity is 100.95. It fits within the range. Then you have got the number of heavy atoms, 20. Again, that fits very well. I'm so sorry. Very well in what he has said. The molecular weight is in between. Yeah. And then the fourth parameter is log P. Again, log P is between 2.11. So sulfasalazine also passes with all like flying colors this filter as well. Right. So again, the point is, what I'm trying to make a point is, none of these filters are perfect. You will never get a filter which will clearly distinguish a drug from a drug like molecule from a non-drug. But then when you try and add multiples of this, you eventually will catch some molecules which were passed by one filter, but then they did not clear the other filter. So that's why using multiple filters will solve the criteria or the problem. Right. So so then you may say that okay, this is all fine, but how do I calculate these properties which will tell me okay, this molecule is good, this molecule like is drug like, lead like, or something like that. There are many things, out of which the one which I would recommend, and many of you probably are already aware of this, SwissAdmi. So if you look at the black strip here, the menu item, there are other parameters as well. Swiss bioassociates. So if you've got a problematic substructure or functional group in the molecule, so you can look for a bioassociatic group. And at least students, as a student, if you have studied bioassociaterism or isosterism and QSR, you should try out these simple things with simple drugs, and God knows you actually understand the subject very well, and you might actually come up with some out of the box solution, because you guys are so smart. Okay, so having said that, in case of SwissAdmi, all you have to do is just create a smile string for your structure, and eventually put it here, and it will predict all the properties, all drug-like properties, pharmacokinetic properties, metabolism, and it will predict whatnot, lots and lots, apart from what we just discussed, which are potentially useful in the drug-like filter, right? Another few slides, like 2-3 slides, and we are done. Now this one is very interesting. Drug Lito. This is developed by NIPER. If you see the website here, niper.gov.in, and this is a standalone version. So you essentially can download this tool. It's open source tool, and all you have to do is, this tool will ask you to supply, it will ask you either a mole file of the structure or a SDF file. So if you've got like a collection of 10,000 molecules in the form of an SDF file, and you want to screen a library of 10,000 molecules, could be virtual molecules, could be real molecules, could be a natural product library, anything which is available, or could be a commercial database where the compounds are for sale. So all you have to do is just put that SDF file in this tool, and then it has got, apart from what I have discussed, 10 other different filters. So if you see here, Lipinski's rule is there, MDDR-like rules, Weber rule, Ghosh rule. Yeah, the spelling, that was a typo. It's actually GHOSH. GHOSE, Ghose filter, the same guy whose name stands as the first and corresponding author probably. BBB rule, BBB is for blood-brain barrier. Now, when it comes to BBB, for many drugs, if your target is, if a peripheral action of drug is desired, so you can always fine tune the BBB penetration of molecules. Just the way, if you remember the cetirizine case, cetirizine does not penetrate CNS, and that's how it is a non-sedative antihistamine, because cetirizine contains a COH group and all the acids are prevented from entering in the brain. And that's exactly what we did in case of sulfasalazine, where we wanted the sulfasalazine derivative to go and penetrate the blood-brain barrier. So there are rules, there are filters, there are tools available for predicting whether a compound is likely to cross blood-brain barrier or not, right? So this probably be very useful for the new molecules, NCEs, but then many chemistry teachers can use this tool for explaining the usefulness of these filters in medicinal chemistry, and that's exactly what I want to encourage. If you are not already doing it, teachers, please do so that the students will develop interest in this. And in the lockdown period, when we cannot meet physically, you can have some practicals based on these simple things which is accessible to everyone. You don't really need a fancy software for this. Apart from that, there are many commercially available softwares which will help you in doing these things, but then they are like very costly. So the last slide, or second last slide, what's the take home message? Okay, so drug-like or lead-like filters are very important in medicinal chemistry. Ask a medicinal chemist and he or she will tell you the importance. No filter is perfect. This is a universal truth. Yeah, but they contribute in achieving perfection in medicinal chemistry and drug discovery. Again, this dialogue from what is this movie, Satyamev Jayate. कोई भी देश perfect नहीं होता है, उसे perfect बनाना पड़ता है. So कोई भी filter perfect नहीं होता है, उसे perfect बनाना पड़ता है. Meaning you use many of them. I love movies a lot, so don't worry about my love for movies. But then it always helps students to connect to what I'm trying to explain. So it helps a lot. Perfect. So anyone can come up with their own filter. So the students, this is true with you as well. You can propose some. You might have some naive idea. Go work on it, and now there are so many artificial intelligence or machine learning tools available. Do it and prepare your own filter criteria, and you will be known after Lipinski with your own name, your family name, your father's name, or your combination of your mother, father's name, whatever you want to give, call that filter as. And as long as you can show its scientific vigor and utility, you will that will be taken note by the pharmaceutical industry. Hope you enjoyed, learned something at the end of this webinar. And if you have questions, I would love to answer those.
Okay. Thank you so much, sir. Actually, it was thoroughly an enjoyable and wonderful presentation. Your journey with emojis and sulfasalazine, it's different. It's just not just benzene rings.
Yes.
So there are a few questions in the chat box.
Mm-hmm.
So first question is, what is the percentage of adverse events, the cutoff, such that the drug has to be withdrawn from the market?
Like I said, every drug is different. So ultimately, you have to prove that the drug is more beneficial than the adverse effects it causes. So on an average, every drug has got around 70 different side effects. So in fact, those who are like the statisticians would understand, or they would in a way relate to this. So the moment you have got a statistician is involved right from the beginning when the clinical trials are designed, for this very reason that ultimately you have to prove to the FDA that it is indeed beneficial than the harm it can cause. There's no drug which will be completely devoid of side effects. So as such, there is no cutoff or something, but it's just that you have to prove that the effects of the drug are not merely by chance, and indeed it has got some effect.
Thank you.
So,
Thanks, Pradeep.
yeah.
Correct. one more question in the chat box. So, what could be the optimal log D value for an orally administered drug? 1 minute, sir. Yeah, thank you.
Yeah.
Another, what could be the optimal log D value for an orally administered drug? We normally talk of log P. So, log D.
Yes.
Yeah.
Log D 7.4 between 2 and 3 is good enough. It can have slightly more as well, but in no way it should cross, ideally should cross 4 actually. Otherwise it becomes too lipophilic and all the other problems like toxicity starts popping up.
Yeah. Okay.
Yeah.
Now there
Yeah.
is a question in the chat box by Dr. Bandu. So,
Mhm.
how much physicochemical property predicted using molecular modeling software is actually
Mhm.
useful for comparing with experimental data for drug discovery?
Okay, great question, Dr. Bandu. I would say that before I answer that question, in 2017 I went for a Gordon Research Conference in US on computer-aided drug design. So there I actually met many people which came from the molecular modeling or computational chemistry groups of different companies like Eli Lilly. In fact, one of Dr. Coutinho's student who heads the in silico predictive modeling group in Eli Lilly in Indianapolis. So I did talk to him, and he said that every big company like Merck, Eli Lilly, AstraZeneca, anybody for that matter, they have their own software tools available for predicting the physicochemical properties, and those proprietary tools which are based on their own data, because the data is accessible, like they have data available for lots and lots of compounds, like few millions of compounds. So then the prediction accuracy is much higher, as opposed to if you and me are do this based on what is available in the literature. We will always have issues with the predictability of the model, because we probably do not have access to the very accurate and very high quality data in large number. So in a way, if I have to use a tool which is known in the public literature, or maybe commercially available tools, so they keep on modifying their models every now and then, because they get access to the data. They actually purchase the data and eventually develop their models. So free tools will always have issues of predictability, whereas the proprietary tools will be much better in terms of getting the meaningful or rightful predictions. But nonetheless, even the free tools, they work very well, and some people are so kind and so nice. They keep on modifying these, making them better and better by adding this, like updating the models every now and then. In fact, ADME is pretty straightforward. The predictions are quite accurate. But when it comes to predictive toxicology, if you have to predict the toxicity endpoints, that's where it's kind of little tricky, because toxicity is very complicated. At least for pharmacokinetic endpoints, say human intestinal absorption or PGP efflux, we know a lot of things pretty, even the metabolism in the intestinal wall, we pretty much know a lot of things actually. But then when it comes to toxicity, the toxicity models, many toxicity models are not really accurate, but the proprietary ones, they are actually. So it's an open-ended question. The answer,
Yes.
no definitive answer could be given, but then I hope, in case you want some more literature, you can write to me, and I'm going to send this presentation to the organizers, so that they can distribute it to whomsoever wants it. So you can have this presentation, and which will probably help you in understanding it, or build up in the subject further.
Thank you so much for all of it, the presentation, the whole, making it as simple as possible for students. It was a very complex topic for sure.
All credit
But
goes to
it
Dr.
was-
Rashmi, actually. Believe me, the number of participants were still. Thank you so much, ma'am, for kind of invigorating their
Thank you, sir. I
Yes.
am so overwhelmed by you quoting me every now and then.
yes,
My
yes.
day is successful.
Yes.
No,
You
I
ensured.
meant it actually. Yes, yes. I meant it. I mean it actually. It's not just for the sake of saying it. It indeed
No,
was
thank you,
very,
sir.
very. Yeah.
Yes, I'm sure it was not just the chemistry and the phytochemistry teachers, but I'm sure everybody found it very practical for the students as well.
Yeah. I
So,
just wanted to make sure that the third year student, fourth year is still okay, but third year student should
yes.
who had probably just learned medicinal chemistry in first semester of third year. So
Yeah,
yeah.
you've
So
given them food for thought as well as,
yes, they
given them
can build
something to
upon.
do sitting at home when you have the time.
Yes, yes, yes. There's so much
So,
to do, and believe me, these
yes,
guys are so smart. We are nowhere
I
near
agree.
them, actually. Yeah.
So bioinformatics softwares, you just have to teach them, write down the outline, and
Yes.
they can take it further. You don't have to teach the
Yeah.
rest.
Yes, yes, yes. They
So
will definitely. Great.
I think Abha ma'am wanted to say something.
Yes,
Yes, ma'am.
Yes, ma'am.
Dr. Prashant, I'm a pharmaceutics person, but I understood, I think 80% of what you have said.
Thank
So
you so
this is
much,
a good achievement. And it
ma'am.
was
Thank you so much.
really, really very good. Means all the informations and I could absorbing. So thank you very much for first for accepting our invitation. And it's really means I think a student must have understood 100%.
Yeah,
Those,
yeah, of course.
yeah,
I hope. I mean, that's the purpose.
Yes.
If if the participants don't understand it has no meaning other. I mean, it's just a waste of time of everyone actually. So that's why I just wanted to make sure. Yeah.
thank you.
Yes.
Thank you, doctor.
Thank you so much, sir. We
Thank
never met. Of course, I heard about you
Mm.
all the time, but I never had a chance to meet you in person. But thank you so much, sir.
Yes. Okay. So thank you, everyone. We have shared the feedback link in the chat box of Google Meet, YouTube, as well as it will be there in your Telegram group. It will be open till 12 noon tomorrow. That's Friday, 24th. And please fill it for the certificates. You have to show your presence. So today we conclude this day 2 of the ISDP from Met IOP, and see you all again with 2 more interesting topics of phytopharmaceuticals and clinical studies. So thank you one and all, and we conclude this session for today.
Thank you so much, ma'am. Thank you so much.
Thank
Bye
you,
bye.
ma'am. Thank you, sir. Bye.
Thank you.
So now we request all the students to leave the meeting if